Moving on, we have with us Dr. Anuradha Bishnoi, Assistant Professor, Department of Dermatology, Venereal and Leprology at Postgraduate Institute of Medical Education Research, Chandigarh, India. She would be talking on assessing expression of immune checkpoints, including membrane-bound and soluble programmed death on receptor and ligands in vitiligo, and correlating these with serum levels of interleukin 2, 7, 15, and vitiligo disease severity.
A very good morning to all of you, and I would like to thank Professor Davinder Prasad, Professor Hamzavi, and Professor Samia Ismail for giving me this opportunity to speak on this platform. So, because of the time constraints, I've reduced my talk, and I would be talking about the expression of programmed death 1 and its ligands on various T cell types in the blood of vitiligo patients. So, first of all, we should note the interaction between the cytotoxic T cells and antigen presenting cells. So the first signal comes when MHC on APCs interacts with T cell receptors on cytotoxic T cells, and this is the primary signal. To augment this, we have co-stimulatory signals in the form of CD28 and CD80 and 86. But to inhibit this, we have co-inhibitory signals in the form of MPD1 and MPDL1. And this interaction is very important to stop overstimulation of cytotoxic T cells. And this is what is actually utilized when we are using programmed death inhibitors. And when we see this picture, in the center we have T cell, the cytotoxic T cell, and we can see that multiple other cells can modulate this cytotoxic T cells via interaction of PD1 and PDL1. And it is also clear that PD1 is expressed on the T cells, whereas PDL1 can be produced by multiple cell types. We can see that we have this antigen presenting cell here, and we have tumor cells here. And this is what is targeted in immune checkpoint inhibitors. So when we inhibit this interaction between the PD1 and PDL1, and stopping this interaction basically unleashes T cells. So the activity of cytotoxic T cells is increased and they kill tumor cells. However, if this inhibition was to take place when the interaction was supposed to be happening between Tregs and cytotoxic T cells, this would have an adverse impact on autoimmune diseases like vitiligo. So, vitiligo is one of the most common cutaneous toxicities reported with the use of immune checkpoint inhibitors, and this is the latest article in JID by the group of Dr. Quatra. And this interesting article, which was published in Nature Scientific Reports, so what they did was that in PMEL mouse model of vitiligo, they injected MPDL1. And this basically led to repigmentation, and we can see in the second photograph. And this actually correlated with increasing Tregs in the mouse. So in this particular study, our objective was to just see the expression of PD1 and PDL1 on 3 T cell types, that is cytotoxic T cells, Th1 and Tregs. And we used flow cytometry for this. So coming to our flow strategy. So in this figure, with the help of side scatter and forward scatter, we identified lymphocytes. These are the lymphocytes. And then they were further gated on the basis of CD4 and CD8. The CD8 cells, they were further gated on the basis of PD1 and PDL1. And these are the cytotoxic T cells. And we can see that these particular cells, the dual negative T cells, they predominate. And when we take CD4 cells, they were further gated with the help of CXCR3 and delineating Th1 cells, and they were again gated with the help of PD1 and PDL1. And the CD4 cells, which were positive for both CD25 and FOXP3, were our Tregs, and then they were, and this arrow here points towards the dual positive cells, that is Tregs, and they were again gated with the help of PD1 and PDL1. And this is an important table here. So all the results of this study are in this table. First of all, we will focus on TC, that is cytotoxic T cells. And what we see is the number of cytotoxic T cells in patients was significantly increased as compared to controls, and that is sort of expected. And the trend of the expression of these molecules on the cells is important because I could not find in literature a very significant information on this. So when we see TC and TS, that is cytotoxic T cells and helper T cells, the dual negative cells predominate. So most of the cells, they do not produce either PD-1 or PD-L1. So that is the one important thing. And the second most frequently found cells were those expressing PD-1. And that is sort of expected in an autoimmune disease because these markers are compensatorily produced on the cells in order to limit the interaction of the immune cells. The most important finding in this study was the expression of PD-L1, and this was actually found to be increased significantly on cytotoxic T cells in healthy people, but was found to be significantly reduced in people having vitiligo. Same findings were seen in helper T cells, and when we come to Tregs, the PD-L1 expression was found to be significantly increased in people who had stable disease. And so the expected observation was that maybe the PD-1 and PD-L1 were reduced on TCs and Tregs, and this interaction was not happening and T cells were overactivated. But what we observed was that PD-1 was actually similar in patients as well as healthy people, but PD-L1 was increased on Tregs, and it was significantly more in people who had achieved stability by almost 10-fold. So this was probably a compensatory response by people who had achieved stability and was not seen in people who had active disease. And another important finding was the interaction between cytotoxic T cells. So these cells are normally thought to produce just PD-1, but what we saw was that a significant number of these cells were producing PD-L1. And this kind of interaction is important because when two cytotoxic T cells interact with each other, they can inhibit each other, and this interaction was significantly more in healthy people, but not in people with active vitiligo. And this important study, again, in Nature Communications, so there they saw that although the number of these cells is very less, but they sort of determine the outcome in melanoma patients. So when cytotoxic T cells have more PD-L1, they are a poor prognostic factors for people with melanoma. So I'm beyond time, so I would like to stop there. Thank you.
Any questions? Yes, Deepat.
This is a very interesting talk because we have been challenged to understand what's going on with our melanoma patients and the ones who develop depigmentation, and we've thought that there are different processes. The one thing that's also been described recently is that the response to anti-melanoma therapy is partly dependent on the microbiome, gut microbiome of the candidate, the individual who has melanoma. Do you have any thoughts on linking the microbiome to the expression of these PD-L1s and these various inhibitors you're referring to?
Sorry, sir. Microbiome and PD-L1, I have not read about that.
So the microbiome and the expression, are you familiar with any variations?
Pardon, sir?
The microbiome, the gut microbiome and expressions that you've mentioned, do they differ in people based upon the microbiome?
Sir, I'm not aware about the expression of PD-L1 depending on the microbiome.
Just to comment on that, I was also very impressed by the result published in, I think it was in Science, on melanoma and microbiome. And very interestingly, we found exactly the same type of microbiome in vitiligo patient as in our study, but also a Chinese group find exactly the same Bacteroides and Firmicutes ratio in the gut of vitiligo patient, and it's the same as the good responder as to immunotherapy. And it's also the same also in the mice model of Caroline Lepoul. So it's interesting to find it in several ways.
That's fascinating.
Thank you very much.